Hi, my name is Luke Matvey. I'm a master's student at the Auburn University Shellfish Lab, Dauphin Island, Alabama, War Eagle. And here I'm here to present to you some of my master's work so far, which pertains to seabirds and off-bottom oyster aquaculture. So thankfully, unlike most other types of finfish aquaculture, we're not so worried when it comes to birds about the birds poaching our crop or eating it. Thankfully, we got nice hard shells in our oysters. But unfortunately, what we're seeing is when you have all that gear out at your farm site, you're offering a lot more surface area for birds to land on. And so, at least in the Gulf, our two most prominent methods of culturing are off-bottom types, either the long line or the floating cage. And both of these types of gear require a lot of hard substrate at the farm site, whether it be pylons or the pontoons that are on the cages. And so these provide ample opportunity for the birds to sit and roost on. And while they're roosting on the cages especially, I'm sure a lot of y'all see bird poop on the pontoons, and this has caused reason for concern, and regulatory agencies have now begun to try and figure out what kind of risks these birds might be bringing to your farms. So the most notable pathogens that people recognize when you talk about the risks of eating raw oysters are things like Vibrios or E. coli and noroviruses. But another potential pathogen are Campylobacter, and Campylobacter are a species of bacteria which cause severe gastroenteritis. It's going to give you really bad food poisoning-like symptoms. Not really fun. It hasn't really been too much of an issue until recently. In 2021, there was an outbreak cited by the Rhode Island Department of Health, in which they were able to link an outbreak of Campylobacter back to harvested oysters from this little place called Potter's Pond in South Kingston. The Department of Health cited the source of the Campylobacter back to the seabirds. The big issue with that is that we don't really have a good understanding of how this Campylobacter is getting into the oysters. We don't know what the source is of it, whether they be anthropogenic or natural. And so part of the report cited the nearby roosting seabirds as the potential suspects. However, it's important to note that the report didn't actually go and screen those birds for Campylobacter. So what this led them to do, the National Shellfish Sanitation Program now mandates, as most of you know, that farmers must have in their operational plans something to address the birds. They're not explicitly worried about if it works or not yet. You just gotta have something. And they recommend doing this by implementing bird deterrents, of which there are three main types. There are tactile, which provide a physical barrier to prevent the birds from actually landing on your gear. There's auditory deterrents, which use loud noises like air cannons to scare them off of your gear. And then there's visual deterrents, which would mostly use that predator-prey interaction to prevent the birds from landing on your gear in the first place. Unfortunately, this is kind of new on the cusp idea of keeping birds off your gear, so a lot of these deterrents haven't really been tested explicitly for their efficiency, and so we don't really know what type of deterrent might be best for what species of bird you're seeing at your farm, or if there is different types of deterrents that are better than others overall. And so that was the first objective of my thesis so far, is to try and test the efficiency of a simple, inexpensive, non-lethal bird deterrent that we made in-house. Thank you. So we're trying to test our own simple, inexpensive, non-lethal bird deterrent. And our second objective was to determine the prevalence of the Campylobacter bacterium in the seabirds that we're seeing at the farm. And then our third objective was to to test the prevalence of the Campylobacter we're seeing in the oysters that were growing at that same farm site. So to do this, we had to field a design where we set up two cage lines about 40 meters from each other. Each of the lines had 3 cages on them. As you can see on the top is our treatment cages, which have the deterrent on them. That's marked by that little like TV antenna looking thing. And then the bottom cage is our control cage, which is set up as any normal industry would set up their cages. Each of these lines were set up in parallel to a row of pylons, and on the middle pylon facing each line is a camera, which is set up to take photos at 30-minute intervals so that we can monitor and quantify and speciate the birds that we're seeing interact with those cages. So on the bottom right here is just an example of what our deterrent looks like. It's nothing too fancy. It's just a plastic lattice. It's a diamond 9mm mesh bag that we cut up and fitted over the pontoons. And then we took some plastic zip ties and we cut them to a point. They're each about 4 to 5 inches long. and we just place that over each pontoon on these 3 cages. And so on the right there is a example of what the camera photo data looks like. So the top is the deterrent cage, and the bottom are the control cages, and you can see those cameras are set up to keep all 3 cages in the field of view at any given point of the day. Our second objective, we determined how much Campylobacter we're finding in the actual birds by collecting fecal matter samples, which is a fancy way to say we just scooped a lot of bird poop. Real elegant stuff. But basically, what we tried to do, we aimed to get about 9 bird samples every week. That was kind of an arbitrary number. We just decided that that was a feasible amount of bird poop for us to scoop in 1 week. This sampling was conducted opportunistically, so we were relying on there being good weather enough for us to get out to the farm, and then nice enough weather for the birds to actually be at the farm. So it's kind of a double whammy. And what we did with that is we would drive up on the boat, identify a single individual, so let's say a brown pelican or something, try and speciate it as well as we could. And then as we approached it and got closer, we'd scare it off and immediately go and check whatever it was sat on, whether it be a pylon or a cage, to see if there's any fresh fecal matter there. And we defined a fresh sample as anything that was still wet, or as some of my colleagues like to think about it, glistening. And we would collect those in 1.5 mil tubes and resuspend them in the lab. As you can see in the bottom right there, some weeks they were a little colorful, so it's always a surprise to see what you're seeing out there on the farm. After we brought it back to the lab, we would plate it on a chrome auger plate, which basically just turns different colors if the bacteria we're looking for is there. And so we have a Campylobacter chrome auger, which is made to isolate the Campylobacter species bacterium that we're looking for. You can see that in the bottom left, those little red colonies. And then we would take those and we would confirm them, with our multiplex PCR and Sanger sequencing PCR. And that's basically what that gel is showing you right there, that we're getting some positive samples. Our third objective was to evaluate the presence of Campylobacter in the oysters that we're growing. So in that same field design that we had with the 2 lines, we had oysters packed in those cages, and every 2 weeks we would go and sample 12 oysters from each cage. And we would take those 12 oysters and homogenize, which means we basically threw them in a blender. We shucked them, threw them in a blender. And as you can see in the bottom left is an example of that. Looks like some real good stuff. Smells something else. But you'd take 12 of them, homogenize them into one sample, so that you'd end up with 3 homogenized samples from the deterrent line and 3 homogenized samples from the control line. And then we would enrich those, incubate them, we'd plate them on our chrome auger, and then we would test those putative isolates, confirming them with the multiplex PCR and our Sanger sequencing. So here's the results from the camera data. It's not quite in the screen all the way. Oh, it is on yours. Okay, perfect. So you can see here, we have 5 months of data analyzed so far from July to November of 2023. And across every single month, the little star on the top means that there's a significant difference between the volume of birds interacting with a control cage in blue compared to the volume of birds interacting with a deterrent cage in orange. That 6th column there on the end is the total average across all 5 months so far. And as you can see, there's about 1/5 the amount of birds interacting with the deterrent cages there in orange as there are with the control cages on average. And so we can see that our deterrent cages are having a significant effect on limiting the amount of birds that are visiting those control cages that would have otherwise visited those cages without the deterrent. And on the right there, you can see an example. These are 2 photos taken from the same time on the same day, and there's a quite obvious difference in which cages those birds prefer. It was important to note that, as you can see on the top there, that is our deterrent line, and there are still birds there. We used plastic zip ties, which obviously had some give to them, and those bigger birds, like that pelican you can see there, if they really want to sit on it, they're going to sit on it, you know what I mean? And so really, what we like to emphasize here is that there was an effect to having it in general. It's not going to be 100%, but it's better than nothing. Here's a table of just all the species of birds that we're seeing at the farm. So as you can see, in that same 5-month time period so far, we've had 15,000 individuals speciated on the farm. There's 2 big groups here I want to notice. There's the tern species group and the gull species group. We got so many species of terns. We got about 5 that I can knock off the top of my head, and then there's a bunch of others that I have a lot more difficulty, thanks to people at the lab for helping me identify some of those. But I just condensed them here on this table, so it's a lot more nicer to look at, otherwise it'd be way long. And so for simplicity, I condensed them. As you can see, there's a varying difference between the amount of birds of each species that we see at our farm site and the preference for each species for what type of cages they like to sit on. This data here is specific to our farm site, so it might not be the same exact data that you have at your farm site, but it is important so that we get an idea of what we're looking at. So here is the fecal positivity results from those samples that we collected, and we saw from about 220 samples so far a 7.35% positivity rate. This sampling was opportunistic, so we couldn't make any specific conclusions about per species. Here is the results of our our oyster sampling, excuse me. So this is the Campylobacter that we're seeing in the oysters. Keep in mind that this was a enriched assay, so the presence of a single Campylobacter oyster in any of those 12 originally shucked oysters could lead to a potential positive result. So these aren't necessarily a clear, accurate representation of the risk at your farm. It is just what is present at the farm. So we also weren't as pleased with the results from our 16S sequencing. We would have wished that it had been more specific, so we weren't able to identify all the way down to the species level, and we can't make any concrete conclusions about the pathogenicity of the species that we're isolating. So these might not even be Campylobacter that would make you sick. They could be some other species which might not be harmful to humans at all. And so in conclusion, we were able to see that our inexpensive bird deterrent was effective. It was about 5 times less as birds on the deterrent cages as there were on the control cages. Our fecal matter sampling turned positive results for Campylobacter about 7.35% of the time. And our oyster enrichments, which grew Campylobacter, were all from control cages. So if I can go back, sorry, I didn't notice that. On the left there is a table that shows our 6 sampling dates that we've analyzed so far. And as you can see, all the positive results that we've found have been from the control cages and not the deterrent cages. So in the future, we want to continue sampling birds out through late spring, and hopefully we can round out our data set to be a nice year-long photo of what kind of birds we're getting at our farm site. We'd also like to develop a better method to speciate the Campylobacter that we're isolating so we can identify what kind of risk might be at the farm. And then lastly, we'd like to test the efficiency of different deterrent types to figure out what kind of deterrents might be most effective to certain species of birds so that we can inform y'all on the best deterrents for your farm site. Thank you all very much. If you have any questions, I'd like to thank everybody at the Shellfish Lab and Tarnecki Lab for helping me do this research.
All right, thank you, Luke. We're gonna give somebody else a chance here, Tony. Anybody else have a question?
Oh, I was like, not me.
Hey, I just had a question about the substrate that you pulled the bird poop off of. Was it only pylons at the farm, or did you get any off the pontoons of the cages itself, or?
We have a mix, so the majority of the samples we're getting are from pylons, but we do sample off cages as well. It's just whatever we can get out there on any given day.
You said it was opportunistic, so I was just curious. Cool, thanks.
Any other questions for Luke? Here you go, John.
Very nice presentation, Luke. Appreciate that. The one question I had listening to you and seeing the photos, which were really helpful, is obviously if you've got the deterrent and a control, you're giving the birds the control one to land on. And I was trying to advise, how would you do something that would take that away and be able to compare what you've got? Have you given that any thought? So that there would be no, I mean, you've got something with the deterrent ones, and then right next to it you've got the nice controls. So is it a way that the birds could, I can't stay there because it's a deterrent, but look at this, I got a control flat spot over here. Is there a way to approach that? Thank you.
Also,
if you understood that.
For my study so far, we're really trying to figure out the effect of the deterrent on mitigating the risk of bacterial contamination of the oysters. And so in order to do that, we had to make sure that our deterrent line was far enough away from our control line that we didn't have any bleeding over of birds that might have landed on the control line, them pooping in the water, and maybe it disperses to our deterrent line, and that would give us some false positive results on the deterrent line. So right now, we have them pretty spread far apart. In the future, I would like to see if we could have maybe the density of deterrents might not need to be as high as some of us might think. Maybe you could have 1 in 3 cages have a deterrent, and that's enough to cover those 3 cages, or vice versa. So that's something we'd like to look at in the future.
Okay.
All right, thanks, Luke. Another round of applause for Luke.